This is Dr. Rosario again. In the previous video, we discussed the structure of microbial genomes and how they are copied through DNA replication. In this video, we are going to focus on how the information stored in DNA is accessed and used by cells. We have two main learning goals for this video. One is for you to be able to describe the process of transcription, and the second is for you to describe the process of translation. Recall from our previous lecture that DNA is a nucleic acid composed of chains of nucleotides containing the sugar deoxyribose. There are 4 different nucleotides in DNA: adenine, guanine, cytosine, and thymine. In cells, DNA is in the form of a double helix. Each piece of DNA is composed of 2 complementary and antiparallel chains of nucleotides held together by hydrogen bonds that form that double helical shape. We are going to talk a lot about RNA in this video. Like DNA, RNA is composed of chains of nucleotides. One major difference, however, is that the sugar in RNA nucleotides is ribose. Attached to the number 2 carbon in that sugar, there is a hydroxyl group. This is what makes it different from deoxyribose, and you can see that circled here in our figure. Also, like DNA, there are 4 different nitrogenous bases. There's adenine, guanine, and cytosine, which are also found in DNA. However, instead of thymine, RNA contains uracil. You can see that these 2 molecules are very similar to one another. Finally, one of the biggest differences between RNA and DNA is that RNA is single-stranded. This figure illustrates the double helix shape of DNA as compared to the single-stranded RNA molecule. Although RNA does not form a double helix, RNA molecules can have 3-dimensional shape. The nucleic acids can fold up on themselves, and the nitrogenous bases can base pair with one another, forming hydrogen bonds to stabilize the shape of the RNA molecule. There are several different forms of RNA with various functions and cellular processes. Ribosomal RNA, or rRNA, is found along with proteins in the ribosomes. These are the structures that are responsible for actually making proteins within cells. We will talk about these more in detail in a few minutes. Remember, prokaryotes have 70S ribosomes, while eukaryotes have an 80S ribosome. Transfer RNA, or tRNA, is the next type of RNA. These molecules are responsible for carrying amino acids, the components of proteins, to the ribosomes. Not only that, they ensure that the correct amino acid is attached to the growing polypeptide chain. Messenger RNA holds the information for the sequence of amino acids in a protein. Finally, the last type of RNA is regulatory RNA. These are very small pieces of RNA that are involved in regulation of genetic information. They are sometimes called microRNAs, small inhibitory RNAs, interfering RNAs, or riboswitches. We're going to focus our attention on those RNAs that are directly involved in protein synthesis: rRNA, tRNA, and mRNA. get too deep into the details of transcription and translation, I want to take a quick step back. Genetic information is stored in an organism's genome. In prokaryotic and eukaryotic organisms, that genome is composed of DNA. I like to think of an organism's genome as a cookbook. A cookbook has many different sections holding the information for making a variety of foods. You don't cook all the recipes in a cookbook at once. Instead, you only access the information you need for a given meal. Thanksgiving dinner may look very different than summer brunch, for example. Similarly, cells do not utilize all the information in their genomes at a single time. They pick and choose what genetic information or which recipes to access depending on their needs. A meal may consist of several dishes made from recipes within a cookbook. You can't eat a recipe. It is just the information on how to prepare the food. Likewise, genes are just a piece of information stored within the genome of an organism. Genes are the recipes in our genetic cookbook. They hold the information for making a product. Most of the time, that product is a protein. Gene expression is the process of taking the information that is stored within genes and producing that final protein product. The central dogma of molecular biology is a framework for understanding gene expression. Genetic information is stored in DNA. That DNA is used to make RNA, and then that RNA is used to make proteins. This is kind of like making a photocopy of a recipe in an heirloom cookbook and then using the copy to make your cookies. You wouldn't want to risk damaging your grandmother's cookbook by spilling eggs or oil on it. Likewise, by separating the process of making proteins from the DNA molecule, the integrity of the genome is preserved. The step of making RNA from DNA is called transcription. Remember, both RNA and DNA are composed of nucleotides. They're the same molecular language, so to speak. Just like an audio transcript turns spoken English into written English, both versions remain in the same language, but in different forms. In transcription, we're moving from DNA to RNA, but we're staying in the same molecular language. Translation, on the other hand, changes the language, kind of like going from English to Spanish. In cells, we move from the language of nucleotides to the language of amino acids. Expression starts with DNA. That DNA molecule is composed of two strands of nucleotides. It is double-stranded. Only one of the strands is used in gene expression. This strand is called the template strand. In transcription, the DNA template is used to produce a complementary piece of RNA. Since we are focusing on the production of proteins, that RNA is messenger RNA. Keep in mind, however, that transcription can also result in other forms of RNA, like tRNA or rRNA as well. Messenger RNA is used directly in the process of translation. The sequence of nucleotides in the mRNA directs the assembly of amino acids into a polypeptide chain or protein. According to the central dogma of molecular biology, genetic information is stored in the template DNA. Transcription transfers this information to the complementary mRNA molecule. In translation, the information in mRNA is used to make proteins. Since the time that the structure of DNA was first described in the 1950s, this is how we've understood information flow within living things. It is important at this point, however, to mention that not all biological systems follow this path. As we will see in later modules, viruses can be the exception to the rule. What we will do for the rest of this lecture video is to dive into some of the details of transcription and translation. Let us begin with transcription. Here are some of the key things that you need to remember about it. It'll be a good idea to take a screenshot of this slide at the end. Transcription is the process of making RNA from DNA. Before transcription begins, the cell must decide which recipes or genes to access. We will get into some of the details of that later. For now, we are focusing just on what happens once that decision has already been made. Transcription starts at a region of DNA called a promoter. Remember that. Transcription begins at a promoter. The process of matching nucleotides to the DNA template and forming the phosphodiester bonds that make RNA is controlled by the enzyme RNA polymerase. Finally, the last important piece of information is that transcription of DNA into RNA ends at a region of DNA called the terminator. Right? So it starts at a promoter, it's performed by the RNA polymerase, and it ends at a terminator sequence. Transcription is the first stage of gene expression. Here we have a segment of DNA that is going to be transcribed. There are three main sections that I want to point out here. The promoter, where RNA polymerase will bind first. The coding region, which contains the sequence that will be copied into RNA. And the terminator, that signals the end of the target sequence. The first stage of transcription is initiation. This is where the RNA polymerase binds to the promoter region of the DNA. Here, it temporarily opens up the DNA double helix, allowing one of the two strands to serve as a template for RNA synthesis. RNA polymerase then moves down the DNA template. During elongation, complementary RNA nucleotides are matched to the template strand and connected via phosphodiester bonds. Take a closer look at what happens within the RNA polymerase. You can see here that the DNA double helix has been pulled apart. This area is called the transcription bubble. Unlike DNA replication, as RNA polymerase moves forward, the area of DNA behind it reforms the double helix. One of the two DNA strands is being used to direct the sequence of the RNA. This is our template strand. This is very similar to DNA replication. Construction of RNA occurs in the 5' to 3' direction. Free nucleotides can only be added to the 3' end of the growing nucleic acid. RNA polymerase then continues to travel down the DNA molecule. RNA polymerase has three main functions. It temporarily unwinds the DNA, matches new nucleotides to the template, and catalyzes the formation of the phosphodiester bonds that hold the RNA molecule together. This process will continue with RNA polymerase traveling along the length of the coding region until it reaches the terminator sequence. This signals the end of transcription or termination. At this point, The RNA polymerase releases the newly synthesized RNA transcript and detaches from the DNA. From here, we can move to the second stage of gene expression, translation. Translation is the process of making proteins using an mRNA transcript. Ribosomes read the mRNA and use the information stored within it to build a polypeptide chain, also known as a protein. The process starts at a sequence in the mRNA called a start codon. Finally, the last important piece of information here is that translation ends at a region of mRNA called the stop codon. So translation is performed by the ribosome. It begins at a start codon and it ends at a stop codon. Remember that proteins are chains of amino acids. They are connected through peptide bonds. This figure shows the process of joining two alanine amino acids together. You can see that the carboxyl group of one is joined to the amino group of the other. It is the ribosome that catalyzes the formation of this peptide bond. There are 21 commonly occurring amino acids as illustrated in this table. This is what provides the variety and structure of our proteins, right? There are millions of different proteins possible using these different combinations of amino acids. Each amino acid is encoded by a short sequence of 3 nucleotides in the mRNA molecule. These sequences are called codons. There are 64 possible codons. AUG encodes the amino acid methionine and is also the start codon. All protein synthesis starts with AUG. There are 3 different codons that signal the end of translation: UAA, UAG, and UGA. These are our stop codons. The other 61 different codons correspond to each of the amino acids. For translation to occur, we need several different components: tRNAs, ribosomes, and an mRNA transcript. The mRNA molecule, of course, is the product of transcription. The sequence of nucleotides in this molecule will direct translation. Notice that there's a start codon and a stop codon. It is the sequence between these two codons that will actually correspond to the amino acid sequence. Here is the mRNA molecule that we transcribed earlier. I'm going to simplify it by writing it just as a sequence of letters. Now, starting from the 5' end, I'm going to look for the first AUG, or start codon, in this sequence. All right, so there it is. Now, each group of 3 nucleotides after the start codon will correspond to an amino acid. Until we reach a stop codon. In this sequence, the stop codon is UAG. It is the job of the tRNA molecules to carry amino acids to the ribosomes and match them to their appropriate codons. tRNAs have 2 important structural features: an amino acid attachment site and an anticodon that can form hydrogen bonds with a complementary codon in the mRNA. A specific amino acid is loaded on each tRNA before it enters the ribosome. Once at the ribosome, alignment of the codon and anticodon allows for the insertion of that amino acid into the growing polypeptide chain. Here's our amino acid attachment site, and here is our anticodon. Here's our sequence again. Each codon corresponds to an amino acid in the protein chain, except for that stop codon. Now let's talk about the ribosomes. They consist of two parts, the small ribosomal subunit and the large ribosomal subunit. These subunits remain unassembled until they bind to an mRNA molecule. There are 3 tRNA binding sites on the assembled ribosome. The A site is where tRNAs enter the ribosome carrying the amino acids. Remember, A for amino acid. The P site is where the tRNA that is temporarily attached to the growing protein is found. P for protein. Finally, the E site is where tRNAs exit the ribosome. E is for exit. So we have our A, our P, and our E sites within our ribosome. Each one of those places is where a tRNA can be found. Before translation can begin, the small ribosomal subunit joins with the tRNA that matches the start codon. This is called the pre-initiation complex. It binds to the 5' end of mRNA and then scans along until it reaches that start codon. Once the pre-initiation complex aligns with the start codon, the large ribosomal subunit binds, forming the complete ribosome. Notice that the first tRNA is now sitting in the P site. Now a tRNA that is complementary to the codon in the A site will bind and the first peptide bond will be formed. The ribosome will then move down the mRNA, matching tRNAs and forming peptide bonds. tRNAs no longer carrying amino acids will be released through the E site, and the ribosome will continue to move down the mRNA chain. This process will continue until the ribosome reaches the stop codon. Here, the ribosome will pause because there is not a tRNA that is complementary to the stop codon. This will allow for the binding of proteins called release factors that will then cause the entire complex to disassemble, resulting in the release of the completed protein. The details, let's quickly review. In transcription, one strand of DNA serves as a template for RNA polymerase to build a complementary mRNA molecule. Transcription begins at the promoter region and ends at the terminator. The messenger RNA is then used by the ribosomes in translation to build a protein consisting of a chain of amino acids. Each amino acid in the protein corresponds to a codon in the mRNA. Translation begins at the start codon and ends at the stop codon. In prokaryotes, because there is no nucleus, both transcription and translation occur in the cytoplasm of the cell. In fact, these two processes can occur almost simultaneously. We call this coupled transcription and translation. As RNA polymerase builds a new mRNA molecule in the 5' to 3' direction, the ribosomes can directly attach to the 5' end of the growing nucleic acid and begin to translate it, even while it is still being synthesized. Multiple ribosomes can bind to an mRNA molecule in sequence. This forms a polyribosome. Genes can have many polyribosomes attached, resulting in very high levels of gene expression. Coupled transcription and translation allows prokaryotic cells to respond to environmental signals and produce substantial amounts of protein in a very short period of time. In eukaryotes, gene expression is somewhat more complex. Transcription occurs within the nucleus, while translation occurs in the cytoplasm of the cell. There is no coupled transcription and translation. The mRNA molecule must be modified before it's even able to leave the nucleus. There are three major modifications that occur to eukaryotic mRNA. The first is that a special nucleotide modification is added to the 5' end of the molecule. This is called the 5' cap. At the end of transcription, a series of adenine ribonucleotides are added to the 3' end. This is called a poly-A tail. Finally, segments of the mRNA called introns are removed from the mRNA transcript. This process is called splicing. Splicing allows for eukaryotes to produce different mRNA transcripts from a single DNA template. Of course, don't forget that eukaryotes also have different ribosomes. Their ribosomes are 80S, where prokaryotic ribosomes are 70S. Before we go, let's take one last look at the process of gene expression. Remember, an organism has a large set of genetic information stored within its genome cookbook. In gene expression, a single recipe is selected and prepared, building the proteins that are necessary for cellular function. In our next video, we will talk about some of the ways that microorganisms select which gene to express and how much to express at any given time.